Deoxyribonucleic acid, or DNA, is a long polymer of nucleotides, each nucleotide being a deoxyribose sugar molecule, one phosphate group, and one base. Nucleotides are joined together to form the DNA backbone. A second DNA strand then joins the first to form a double helix. You may recognize the code letters A, C, G, and T, which are the 4 bases used in DNA. These bases are adenine, A, cytosine, C, guanine, G, and thymine, T. Like the code in Mr. Gates' software, the sequence of the 4 bases encodes genetic instructions used in the development and functioning of all known living organisms. Once organized, this information is what makes up the gene. And genes, as we all know, contain the recipes for the components of life. Genes hold all the information to build and maintain an organism's cells, and they also pass genetic traits to offspring. Human DNA contains an estimated 20 to 25,000 genes in its approximately 3 billion base pairs. And did you know that if the DNA in an adult human's body were laid out end to end, it would be about 30 billion miles long? That's enough to travel the distance from the Earth to the Sun over 320 times. A gene contains both coding sequences that determine what the gene does and non-coding sequences that determine when the gene is active, as well as thousands of other regulatory functions. Less than 2% of the genome is for coding proteins. The other 98%, which used to be thought of as junk DNA, is now being studied to determine its functionality.
Chemically, the DNA in the cells of my body is the same as that in a fruit fly, an orange, a hippo, an elephant. It's all the same chemically. The uniqueness to all of those species of living things lays in the information content of the DNA. So while the chemistry may be the same, the actual information that's embedded in that chemistry is very, very different. And the challenge for molecular biologists such as myself is understanding completely what that information means.
When a gene is active, the coding sequence is copied in a process called transcription, producing a messenger RNA or mRNA copy of the gene's information. mRNA is produced by a protein that's over 3,000 amino acids long as it reads the genetic code. Wait a minute. How can messenger RNA require a protein that reads the genetic code, but yet the protein needs the messenger RNA to be produced? Could these have both come into existence at the exact same time with the meaningful information needed for both to operate properly? As you can see, much is still a mystery when it comes to the origin of this complex organic language.
And what's fascinating to me, not only do proteins create proteins, but we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences of part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So what came first, the proteins that made the DNA or the DNA that made the proteins?
Once DNA is organized into a single coiled piece, it's then wrapped around a complex hierarchical structure of proteins. This is what is commonly known as a chromosome. There are 23 pairs of chromosomes in the human genome. A genome is a full set of chromosomes containing all the inheritable traits of an organism. And as we observe our surroundings, we can see there are many different genomes in existence, creating very unique and incredible organisms. If it seems like your body is constantly working hard, it is. Living organisms continually produce new cells through a process called cell replication throughout their entire lives. And here's how it works. First, we start with our DNA double helix. Helicases are molecular motors that use the chemical energy of ATP to break hydrogen bonds between bases and unwind the DNA double helix into single strands. This process is used for both replication and repair of the strands. If unwound, a single DNA strand would be about 6 feet in length. And yet, It fits into a cell which can't even be seen with the human eye. Remarkably, these strands are kept from tangling as they are separated and duplicated. Next, these strands are copied by enzyme-based computers, creating an exact replica of each original strand. Over 30 different proteins are required for duplication and repackaging of cellular components during cell division. It's from this tiny, masterfully complex cell that life is formed. All living organisms begin with one cell. As cell division takes place, we see multicellular organisms take form. When we stop to think about our own body having approximately 100 trillion cells, each group having a unique purpose, such as epidermis, the lungs, heart, skeletal structure, muscles, nervous system, digestive system, reproductive system, immune system, and the brain all working simultaneously to form this magnificent machine we call the human body, we begin to have an appreciation for the complexity, elegance, and beauty of life. And as we look around us at the millions of plant and animal species, it becomes incredibly breathtaking to ponder how much information and organization must take place for all of this life to exist.
You know, we're all derived from a single cell, the zygote. And what marvels me is from this single cell, we get 250 distinct cell types, each of which has a very, very unique program. All of the cells, with a couple of exceptions, have a nucleus and have a genome, and that genome is the same across all the cells. So when we begin to think about the complexity of the genome, I think that's amazingly complex, how we go from a single cell to unique cells that have very specific programs of gene expression that are necessary for that unique cell to function. But we're only beginning to understand the information in our genome that directs those unique programs of gene expression.
While we know a great deal about the cell and its functionality, we've only scratched the surface of its inner workings. Scientists are continually discovering new information and data concerning the cell and its DNA. As our knowledge increases, it gives us a deeper understanding of how complex a single cell really is. And as we ponder this miniature marvel, we begin to wonder, could this simple cell have evolved from the elements of the earth over time? Is it possible for this to have happened by natural processes? Well, we're not the first ones to ask this question, and it's a difficult question to answer. In the field of information science, there are rules for probability which help us define and understand the possibilities of an occurrence. So to answer that question, we must first define the terms of probability.